Yeah, okay. So welcome everyone to our seminar. Thanks for your flexibility as we move around our times a bit, but we're back to our normally scheduled time. And today we have another one of our poster prize winners from last year, Yasmin Ben Ishay. And she comes to us with her bachelor's and master's in chemistry from Ben Gurion University, and is currently doing her PhD at Weizmann with Daniela Goldfarb. And she will present her research today on unraveling the dual nature of PpiB in solution and in cell by EPR spectroscopy. So with that, the floor is yours. Oh, Yasmin, you're on mute now.
Of course, I'm going to be on mute. So thank you, Joseph, for the kind introduction. And as you said, I am a PhD student in the Goldfarb lab at the Weizmann Institute of Science in peaceful Israel. And today we're going to unravel the dual nature of PpiB in solution and in cell by EPR spectroscopy. And for the physicist and the chemist in the audience, I promise that I will be that I will walk you through all the biological terms, and then we can then hopefully you can follow me till the end of this presentation. So let's start. So in this presentation, first I'm gonna give a bit of background and I'll tell you about our motivation to perform this study. Then we're gonna focus on the model protein that we chose to work with. I will elaborate a bit about the variants that we generate, then the spin levels that we chose to work with. Then we're gonna go on the result in solution and compare it into the cellular environment. And eventually, we're going to talk about what we learned from this study. Because if we don't have a bottom line, then it doesn't worth to publish. So, okay, so let's start with it. So I'm going to go big now. I'm going to ask the biggest question that biologists are being asked. Why to study protein? Why biologists are so obsessed with protein? And I will give you also a big question, a big answer. It's because they are the key players, literally the main actors of every process in living creatures. So if we want to understand this process, we need to understand the dynamics, stability, structure, and all of them affect the functionality of the protein. And this is the big motivation. In order to understand the protein structure, we need to understand the structural complexity level. And I'm going to do that really fast. I just want that everybody will understand when I'm talking about these biological terms. So imagine yourself as a child. You want to study something that is very, very tiny. So you take a magnifying glass and you go really, really, really close and you zoom in the most that you can. If you're going to do that on a protein, the first thing that you're going to see is the primary structure. And the primary structure is basically amino acid that is conjugated by a peptide bond that forms this chain. If we're going to zoom out one level, then we start to see the secondary structure. And this is mediated by hydrogen bonds. And you start to see these elements, which we call motifs. You have the alpha helix and the beta sheets on the right. And between them, you have loop or turns, depend who you ask. If we're going to zoom out again, we're going to see the tertiary structure. And here we see the 3D structure of the protein. This is mediated by ionic interactions, the sulfide bond, and hydrogen interaction. Another level up, we're starting to see the protein-protein interaction, the 3D proteins that are interact with each other, and this is called the quaternary structure. All of these levels had been established and visualized in many techniques a long time ago, but then we reach to the elusive concept, which is the 5th level. Here we're talking about the quinary structure. It represents the transient protein surface interactions with micromolecules in the cell. And why is it so elusive? This concept had been conceived in the 60s but it is very difficult to investigate proteins inside cells. And as the in-cell approach started to emerge and was implemented in different spectroscopic and microscopic techniques, we started to gain this experimental data in order to support this concept. And this is something that I will be focused in my talk. So remember, quaternary structure, very important and elusive idea. The majority of protein studies had been conducted under artificial conditions, which means in a solution or in a buffer or basically in a test tube. You go to your lab, you do what you want, and you have that. Unfortunately, it does not represent the native environment of the protein. The native environment of the protein is usually the cell, and the cell had in it many features that are pretty unique, like the crowdiness, the thickness, the specific and non-specific binding, the transient interaction with cell component, and all of these effects may cause some change or affect again all of the feature of the protein. If we're talking about dynamics, stability, and structure, and all of these affect the functionality. So if you want to generally obtain an information on the protein, you have to study it in cell. But it's not easy, and let me tell you, it's not easy to study protein inside cells. As it turned out, EPR is a powerful tool to explore protein dynamics and structure. An inherently requirement of the EPR technique that we need to introduce a spin label on the protein usually we're doing it by site-directed spin labeling. And then we can observe or track the structural or the dynamics, depends which technique you are using, of the protein only. And it doesn't matter what there is on the surrounding it, because the EPR is basically blind to it. It is true, we need to highlight here that the majority of the cell compounds are diamagnetic, although there is some species, some paramagnetic species, and the main one is the manganese. But once we know that, and this is only some small obstacle, then it's much more easier to deal with. So from here, I want to extract our objective to this study, and it is to decipher the manifestation of a quinary structure, again, the quinary, don't forget, on a well-structured globular protein by EPR spectroscopy. So basically, we want to use this advantage of the EPR spectroscopy to study protein and to gain more experimental data of the existence of quinary structure in cell. From here, the model protein that we chose to work with is Escherichia coli peptidyl prolyl cis-transisomerase B. This protein regulates the protein folding by catalyzing the cis-transisomerization of the proline-imidic peptide bond. And it usually, proline is a key amino acid because it can be involved in the activity and the structure of folded proteins. So it's an important protein for E. coli. We chose this protein because it's a well-structured globular protein. It does not have these significant conformers. So any difference in the structure could be suggested to be the quinary structure. It was also well-studied protein by NMR and EPR in solution. So we have a lot of data that we can compare with. And as I said, it's an important protein for E. coli. So it's the main cytosolic PpiB. It's also a pivotal chaperone for cell survival and adaptation. And this is very unique and important feature of E. coli, which is a one-cell organism. If you don't adapt, then you just die. And of course, the protein does not, the bacteria does not want to die. And it was also reported to interact with many proteins or cell compounds. So this is great for us because if we expected that maybe we're going to see some difference if we're going to measure it in solution and compare it to the measurement conducted in the cell. The variants that we generate for this study is first the double cysteine mutant, and as you see, we located a genetically encoded cysteine on an alpha helix element, which is related to what? To the secondary structure of protein. Here you see, I taught you something. The second one is located on a loop, and here and with that we can actually compare the two elements behavior. The second variant that we produced was almost the same labeling site, except that here we incorporated an unnatural amino acid, which we call pyrrolysine, or O. From now on then, we're going to call it O. And this introduced a unique linker that actually gain us the ability to label it with different spin labels, because we have different labeling scheme that we can use here. Two things you need to know about the difference between the variants. First, yes, you can ask me why you didn't incorporate the unnatural amino acid exactly as the previous variant, and the reason is because the incorporation of unnatural amino acid is highly dependent on the location in the protein. So you need to produce many mutants, and then you can select the one that is getting expressed really well. And also you need to see that it's stable. And we also verify this by nano DSF. So we know that it's stable. Another thing is that, again, as I said, here you have only one type of labeling, which is conjugated to the cysteine the cysteine residue, and the other one you have two labeling chemistry that we can use. So it was expressed. Great. Now we're gonna talk about the spin label that we chose to work with. These spin labels were chosen either in order to utilize their advantages for our to use in our study, or for a control purpose, only to compare. We're gonna, if you're gonna see on panel C, you're gonna observe all the spin label that can be attached to a cysteine residue. We're gonna start from the right to left, like in Hebrew. You learn another thing. First, we have the gadolinium spin label, the DO3A gadolinium, which was reported to be very stable for in-cell studies and show a high sensitivity, especially in W-band, and this is the spectrometer that we use. The next one is MTSL. It's a commercially available nitroxide spin label. It forms a CS bond. This bond tend to break upon the reductive cellular environment. The second one is M-melamidoproxyl. In shortly, we're going to call it 3-MSL. And this spin label is conjugated with a CS bond. A CS bond is a much more chemically stable linker, which we can utilize for in-cell studies. Both of these commercial available nitroxide, and in general, I have to say, to extract, the nitroxide is getting reduced, and we basically lose the paramagnetic feature, and therefore we cannot see it anymore. The signal just reduces until you cannot see it when you introduce it into the reductive cellular environment. So our star, not ours actually, the star of the this study is the MTET bond. Why is that? First, it formed the CS bond, which tend to be, as I said, is stable inside the cell, and also there is a functional group that shield the nitroxide. This spin label had been was synthesized from our collaborators, the Miller group from Marseille, France, and we wanted to utilize its advantages for the in-cell studies. To the unnatural amino acid, we conjugated gadolinium spin label, as you can see here, via the click reaction. Now I'm going to explain a bit better about the click reaction. So here you can see the unnatural amino acid residue that buries the alkene. To that, we're going to conjugate the azidogadolinium, as we see before, that buries the azide, and we're going to perform, with the help of copper-1, a copper-1 catalyzed alkene-azide cycloaddition click reaction. Eventually, we receive this the spin label that is conjugated to the protein. Now, you can tell me, wait, Yasmin, it seems long and flexible. Hold that thought. We thought about it also, but first you need to see the results and then you can decide by yourself. So let's continue. The EPR technique that we wanted to use in this study is, first of all, it's the expand continuous wave, CW shortly, EPR. We're going to measure it at ambient temperatures because we wanted to, again, see the dynamics, and we want it to be the closest that it can be to the native environment of the protein. We will perform that in order to probe the residue-specific dynamics of a nitroxide. Why nitroxide? Because gadolinium, with gadolinium, you cannot see anything in room temperature anyway. It's too broad. So by following the line shape of the nitroxide, we can understand if it is moving in a fast motion, like here, or in a rigid one. The second EPR technique that we wanted to use is a pulse dipolar spectroscopy, and specifically double electron-electron resonance, or in shortly DEAR. We're going to perform that at cryogenic temperature using our home-built W band, which we really like and is very beneficial for gadolinium, to observe gadolinium spin labels. And we're going to do that in order to probe the structural conformation change and population. So from here, our initial aim was to perform an orthogonal gadolinium nitroxide labeling, labeled PpiB, and then we incorporate it inside cells, and we can observe the dynamics of the nitroxide and the structural conformation or distant distribution, basically, of the pairs that we chose. And in one sample, we can actually get all this information. And this idea was great, but we're going to see how it goes next. And again, here you can see an example that we utilize a different type of spin labels, and we utilize their benefits. And of course, we are using also the EPR techniques to gain a lot of information about our protein. So let's start to see the result. I've talked about our plan. Now let's see the real deal. We first started with MTET singly labeled PpiB. We carry out an CW EPR, and instantly you can see that the line shape is characteristic to a fast motion. Now, we wanted to also examine both of the labeling position. The first one is on the alpha helix, which again, this is more structured. The second one is on a loop, just to give you a brief reminder. So we double labeled with MTETPO. We put it, of course, and here I'm talking about in solution. We started with to examine everything in solution first. And again, as you can see here, a line shape that is characteristic to a fast motion. So from here, we can summarize that the spin label conjugated at both selected position, either on the alpha helix or on the loop, are highly mobile in solution. Next, we wanted to examine the orthogonal gadolinium nitroxide variant. Now, this is where the problem begins. First, we have to say that we managed to achieve the orthogonal nitroxide gadolinium labeling, and we verified the high labeling of both of the spin labels using mass spectroscopy. Then after we performed that, we wanted to acquire CW EPR, and as you can see here, it had changed from the previous measurement. You see here that the motion is relatively rigid comparing to the previous one. And again, here we are talking about the motion of the M-tadpole that is located on the alpha helix. Now, what is the difference between this one and the previous one, and why the dynamics had changed so much? The only difference that was changed here, that here we first performed the gadolinium labeling using click reaction, and then we perform the secondary labeling of the nitroxide. Okay, we're gonna keep that in mind. Then we thought maybe if we're gonna change the spin label, then it will make a difference. So we label with MTSL and again with the gadolinium, and we saw again this rigid limit region that appears. So from here we can conclude that there is something that restricts the motion of the nitroxide, but it still doesn't cancel our aims. So, okay, it's suspicious. We need to bear it in our mind, but we can continue. Next, we interrogate our variants using DEAR. As you can see here, there is the double label cysteine mutant, the double cysteine mutant variant. And we label it with M-tadpole and with DO3 gadolinium only for a control purpose. And as you can see in panel B, we get a narrow distribution line with a maxima at 4.5. Great, that seems the same. Now we're gonna we carry out DEAR interrogation on the variant with the unnatural amino acid. We took the two spin label variant, again, once with MTSL and second time with M-tadpole, and we observe the distance distribution. As you can see here, it's also relatively narrow and it shows a maxima at 4.5. Now you can stop me and say, wait, there is some shift in the MTSL. This we took into account because when you use different spin label, then you have a different linker. It makes sense that there will be a bit of a shift here and there. But from that, we can conclude that the distance distribution does not depend on the spin label. And this is amazing for us. Why? Because we know that every change that we're gonna see derives from the quinary structure and not from the spin label that interact with something. So good, we are all done and we can now continue to the in-cell part. The majority of in-cell EPR studies usually were conducted on a protein that was delivered by the majority, let's say, electroporation to mammalian cells or injected to exopod outside. But both of them is not the native environment of our protein. And our initial aim is to investigate in its native environment. So luckily, our collaborators just recently published a paper where they incorporate a protein, a spin label protein, and to E. coli cells. And they managed to observe its dynamics by CW EPR. So we took their initial protocol and optimized it a bit because we also wanted to explore it using DEAR, and with DEAR is less sensitive than CW. And we continue with that. Just one thing that I would like to empathize, this protocol is relatively very fast. It means that within 20 minutes from the electroporation part till the time that you start to measure, we are talking about here about 20 minutes. And 20 minutes is enough to observe nitroxide that yet been reduced. So actually, it will open you this opportunity to explore protein also using unshielded nitroxide or nitroxide that are not compatible to a biological system. Okay, so we first wanted to make sure that the protein get inside the cell and not connected to the membrane or is present next to it, the vicinity of the cell. So we label PpiB with fluorescent dye. We deliver it inside E. coli cells, and with this we can actually optimize it. And as you can see in panel C, the protein is inside cells and in a quantity that allow us to perform the investigation that we wanted. Another evidence that it's getting inside the cell is if we recorded an echo-detected EPR spectrum. Of here, we are talking about double cysteine mutant that was labeled with gadolinium. The reason we use, again, gadolinium is just because this is easier for us to work with, because we have W band, and also because we don't need to think about it being reduced. It is getting reducted. So as I said, gadolinium is perfect for in-cell studies. From here, you can see the central transition of the gadolinium superimposed on the endogenous manganese. And if you remember that I said before, this is the paramagnetic species that we saw from the inside of the cell. Okay, so we know that protein is inside the cell. Now we can start to interrogate it by EPR. Wow, I'm doing it so fast, I'm sorry. So as I said, we took MTEDPO singly labeled PpiB. We delivered it inside E. coli cells, and then we acquired the CW EPR. As we started the scanning, boom, we saw this huge difference, this significant change between the dotted spectrum, that this is represent the in-cell results, to the solid line that represent the in-cell results. And from here, we can understand that the nitroxide that is located on the alpha helix had changed from a fast motion to a slow motion. Then we said, okay, but we wanted to investigate it, the two labeling location that we chose. So we took the MTEDPO double labeled PpiB and we introduced it inside E. coli. And again, boom, you see this significant change from a fast motion to a slower motion. Somehow, both of the labeling sites are getting restricted. Then we said, okay, maybe the MTEDPO is interacting with something. Let's try a different nitroxide. So we label with a 3-MSL, a single label. So it means the variant with the unnatural amino acid. So we have only one cysteine. We incorporate it inside the E. coli cells. And then again, you see this dramatically change between fast motion to a slow motion. So from here, we can say that the residue-specific dynamics of both labeling locations have dramatically decreased upon the cellular delivery. And that is great because we're starting to see something different. We always thought that the environment, the cellular environment might affect, but here you can actually see that. So it was mind-blowing for us. Then we wanted to follow the reduction of the spin label by time as a function of time. We acquire a CW spectra. Okay, we acquired a CW spectra as a function of time. You can see in panel A the 3-MSL labeled PpiB, in panel B the MTEDPO labeled by PpiB, and in panel C you can see the reduction profile of both of the spin label. From this graph, you can clearly observe that while the 3-MSL loses 80%, again 80% within 60 minutes, the MTEDPO loses only 10%, and again from the initial signal amplitude. So That was great because it just give us confidence that we chose to work with a good biocompatible nitroxide spin label. It is also an evidence that, an additional one, that the protein is inside the cell, because if it was outside, then the 3-MSL will not be reduced as it happens here, as it's shown here. Then we wanted to investigate and to understand the features in the cell that govern this change that we previously observed. So the first thing that we thought is probably it's because the cell is much more condensed. So the crowding effect must restricted the dynamics of the nitroxide. You can think of yourself if you're going to a party and you start to you come early, then you can dance and move and do all the movement that you want. But as the time goes by, people are coming and then it's being crowded, and then you have to restrict yourself and you cannot dance with all of this, with high mobility, let's call it. So the same thing we thought we're gonna observe if we're gonna take the MTEPO double label and we're gonna embed it with a different crowding agent. We also add the charge effect in order just to see if it matters. So here we chose to work to embed the protein with a lysozyme that represent the negatively charged crowding agent, with BSA that represent the positive a crowding agent, and fecal is the neutral one. And again, just by looking on the spectra that we received, you see that we did not manage to mimic the same significant change like when we obtain inside the cell. So from you see some mild change, which suggests mild contribution, but still we can conclude that the crowding agent failed to reproduce the observed change in the cell. Then we thought, okay, we know that PpiB interacts with many proteins inside the cell because it's a chaperone, it's also a PpiAs, it makes sense. So probably it bounds to something. So we investigated the MTEPO double label with cell lysate. As you can see in panel A, the line shape did change. In B, you can see it more clearly between the 10 minutes and the 20 minutes. You see this, again, rigid limit region that start to appear. But if we're going to look on panel A, we see that the main process that govern here is the reduction of MTEPO. Now you can tell me, you can say that you can sorry. Now you can say that I maybe trick you because you see here that the MTEPO gets reduced really fast, but and this is a phenomena that was already been reported previously, and we know it happens, but in cell MTEPO stay stable for a long time. Now this only extract us that the importance of exploring protein inside cells is it's very important. And why is that? Because the cell lysate cannot mimic the cellular environment. So if you really want to generally obtain information on a protein, you need to do that inside the cell. And to conclude this slide, E. coli lysate does not recapitulate the observed change in cell. Now, as I said before, our initial aim was to deliver the orthogonal nitroxide gadolinium PpiB inside the E. coli cell, and then we can observe both the dynamics of the nitroxide and the distant distribution using, again, CW and DIR. But this variant, after the spin labeling, tend to aggregate at high concentration, and this is a requirement that we have to achieve in order to deliver it inside the cell. So now we thought, okay, maybe we are stuck here, but this is not true because we just can reproduce another sample. We don't have to do everything in one sample, but we still can examine the protein inside its native environment. And we can do that. We chose to do that with gadolinium. Why? Because gadolinium, again, you don't need to think about how it gets reduced or maybe even on technical issues like that is related to the nitroxide. So here we continue with the gadolinium double spin labeled PpiB into for the deer interrogation. I just want to highlight something that we noticed that maybe it worth to know. We suspected that the histag that was present in this variant maybe interact with the copper-2. Now you can ask where this copper-2 come from. So in order to perform the click reaction, we had to put a lot of excess of copper because it uses the copper-1, but you still always have an equilibrium with copper-2. And we at the end, we do try to extract it with or to remove it with EDTA, with its chelator, but I guess it did not work so well. In order to, and we thought that maybe this interaction of the copper with the histidine somehow restrict the motion of the nitroxide located on the beginning of the protein, by the way, that is more close to the C-terminus of the protein. And in order to prove that, we perform an echo-detected field sweep, and we just make it as a wide field sweep. And we did see the copper, and we observe also the G perpendicular that's suitable to a copper that is bound by 1 by 2 histidines or more. So yay for us, we had an assumption, we prove it by EPR, and this just show you how EPR is such an amazing tool to investigate proteins. Okay, so we continue to the deer interrogations. So as I said, we take the gadolinium double-labeled cysteine, double-labeled mutant, and we deliver it inside E. coli cells, and we managed to acquire a high-quality deer trace. And this was very nice because, again, working with E. coli is not something that is common, especially with deer. So we were very happy with that. We also wanted to investigate the protein in its non-native cellular environment. So we incorporated it inside HeLa cells, which are mammalian cells, similar to ours, and of course does not contain all the cell components that probably PpiB interact with. And we expected not to see a change. We could, of course, compare both of these deer traces with the one we got in solution. If you look in panel C, you can observe that the result we got from the non-native environment of a cellular environment of PpiB is almost similar. It has a narrow distance distribution that obtain a maximum of 4.5. Now let's compare it to the E. coli one. And here you can see a significant broadening of the distance distribution that have, again, the same maxima, but a broadening. This broadening suggests, implicate that, depicted that we have a higher number of conformation of PpiB inside the cell. And now you can see that it actually connects to the definition of binary structure, because here we are talking about the small differences on the surface of the protein that, of course, is affected by the context, the micromolecules that present in the cell. Again, here you can see it very clear. And this was very nice to see because it's, again, it's correlated to the result we observed in using CW. And both of them just tell us that we actually find an evidence for the existence of the quinary structure for PpiB in its native environment. With that mind-blowing thing, I'm gonna summarize my talk, and I'm gonna I can conclude that PpiB structural behavior differs significantly between the in solution and the in cell. How we got to this conclusion? Because we saw that the mobility of the spin label located on both investigated sites, either on a loop, or that is more flexible, dynamics should be fast, or on the alpha helix that is more structure secondary element, had dramatically decreased in the cellular context. We also observed that broadening in the distant distribution of E. coli depicting the change in the structural space of PpiB. So again, with that, combining the two results, we can suggest a quinary structure of PpiB in the cell. If you want, and we were very happy, okay, to see that. It's really nice. I know maybe for people who doesn't work with biology, to gain some confirm a result, it is really nice, because when you work with biology, you don't know what happened there, and when you understand that you do, it just makes you very happy. So if you want to know more details, you can see our new publication that was accepted in Protein Science, and hopefully it will be published in the upcoming days. So stay tuned, and you can follow our Twitter, and probably it will be published there. With that, I would like to finish and to thank you for listening and for surviving till the end. This is very nice of you. And I will thank my supervisor who advised me and follow me and encourage me through all of this journey. To our collaborators, Shu Cheng Su, that provide us and synthesize the DO38 gadolinium from the Mileo group, that provide us the MTEDPO, the shield nitroxide spin label, and helping us with the initial protocol for protein delivery inside the cell. I would like to thank also the people from our service unit in the Institute, especially from Yoav, who I just hijacked his office in order to give this talk. To my lab members, Arina, Elena, and Alexei, to our former lab members who we miss very much, Manas and Maria, to Professor Gottfried Otin and Mitun for introducing me to this wonderful world of unnatural amino acid incorporation and a click reaction, which it just give us an endless possibility that we can do with biology. And of course, to Inbal, Gilad, and Vital, who helped me to establish this system in our lab. And with that, I'm gonna shut up, and you can ask me whatever you want. Another thing, I would like to thank the EPR community just for supporting me and sending me message through this difficult time. Your support is highly evaluated, and it really encouraged me. So thank you. And I'm happy to answer any question you have.
Thank you for such a wonderful talk, Yasmin. See some clapping hands. Don't get tired of that emoji. Okay. Marlene, you've raised your hand. You can just unmute and speak. Oh, the unmute part is important though.
We cannot hear you.
Still no audio.
Is it better now?
Yes,
yes, we can hear you.
Yes, I'm sorry. Thank you very much for your presentation. It's a very interesting work. I actually have many questions, so if someone else has questions, I can just stop talking, and so you let me know. I was wondering if you actually test the presence of copper in the activity or the function of the protein, and because usually you didn't mention that, or maybe I missed it. I was wondering if it was not deleterious for the function of your protein.
So we actually did not check the activity of the protein because this is a mutant that we already been working in the lab previously, and we saw that it's active and it's performing all of that. So no, I haven't checked it. And
But
I
even
did
in
check.
presence of copper, is it active in presence of copper?
You mean if the copper is somehow affect its activity?
Yes.
So no, I haven't checked it. I don't think it should have some effect on the activity. This is something that we plan to because we had some problem, then we wanted to understand what happened there, and this is another step that we can see if, because once you lose the activity, then it doesn't matter what you observe.
Yes, that's why I was asking.
Yeah. So no, we haven't tested that. We are planning to do that. The reason that we haven't done it is just because we did not have any test that we can perform inside cells. And this is, I mean, I can perform whatever I want in the solution, but if I don't see it like in cell, then it's also a problem. So
yes, but at least in vitro, you know, if you lose the activity in vitro in presence of copper, for sure in cell you will not have activity. So
yeah,
maybe
but
it's
first,
worth it at least to do it in vitro.
No, so I did not perform that. But if we really want to do that, then we need first to get rid of the HISTAG, and then we can continue because now we know that the HISTAG is a problem because it just bound very strongly to the copper. And the thing is that in order to get rid of all the stuff from the click reaction, I do many washes. I first use a desalting column, and then I wash it for 4 times, only just to exchange it to a DO3-based buffer. So we thought that it will be enough. I guess not. Also, we do a treatment with the EDTA in order to.
Yeah, you mentioned. So, since you are can I continue, Joseph? You tell me.
No, please.
As you were mentioning the HISTAG, I was wondering if in your first EPR, CW EPR spectra, you did have your HISTAG or not? Because in the figure, you don't have it. Yeah, the very first one.
Yeah,
that
so
you meant that you
here.
Yeah, this one. So you don't have the HISTAG here?
No, I have it all in all of
the way. Okay.
Yeah.
So that means that if here you don't have a broadening of your spectrum, you think that the copper, for sure, the copper will coordinate the HISTAG because it's how it works. So you think that when you add copper, it will modify the environment of your label? Is it what you meant, right?
Yes.
Did you try with another metal, like zinc or something, just to put something in the HISTAG? And because why don't you have? I don't understand why you don't have any broadening here if you have your HISTAG.
Why I don't have what? Sorry, can you
the
repeat
broadening
it,
here that you mentioned?
Why
don't you have it here? Because you have your HISTAG.
So we thought that the reason that we see this broadening, or it's more correctly to say the restriction in motion, is because of this formation of the copper plus the histidines there. Here, there is no copper because we did not perform any click reaction. This is why we have to compare to understand what is different between the two variants, and And then we understood that the gadolinium labeling somehow changed it. The first thing that you suspect always is the copper. And again, this about what you said, that this was a really good question, and that you can just add something that will be bound to the his tag, and then you can overcome this formation of this structure of a copper that is bound. And this is something that we are trying now. Just to bound it to, you can use even a nickel beads and just perform that. So to answer your question, we did not really examine the activity, and this is a problem, but we can see that eventually we did not continue with it. So we just wanted to show what did not work, and I think this is also important for people to understand, and then they can overcome it somehow and manage maybe to do that better than I am.
Okay.
Yeah, I hope that I answer your question.
Yeah, more or less.
We can continue.
It's working. I mean, I'm sure it's work in progress, so that's not easy. I have a last question, maybe. It's about the incorporation of your non-natural amino acid.
Yes.
I was wondering. Well, I have two questions in one.
Okay.
I was wondering how much incorporation you have, how much protein can you get after the incorporation of your non-natural amino acid in the protein. And then as a, let's say, a continuation of the question would be what concentration of protein and with the non-natural amino acid you have in your deer experiments.
Okay, so luckily, at the first time that we performed this incorporation, we gain, as you can see here, there is a relatively high concentration of the protein. Eventually, we finish with a protein at, I can give you the numbers. So we had something about almost 3 milliliter of 320 micromolars. So relatively to unnatural amino acid, it was crazy.
Yeah,
this
yeah,
is
it is
something
a lot.
I managed to work. I was optimizing the labeling for a year only on this batch, and this we get it from 1 liter of cultural, which is, I have to say that it's actually, I mean, it's nice that you asked because it's not the right audience, but it actually was amazing to get that. We were very happy. And when we talk about with expert here in Israel that is doing that, he was literally told me that the stars had line just line up, and this happened. So
okay, no, yeah, it's very good. You have a lot of protein with a non-natural amino acid inserted. That's not an easy job. So yeah, that's nice. Very nice. Thank you.
Thank you for asking.
Okay, the floor is open for other questions. If anyone else wants to. Okay, there are 2 hands up. I saw Alberto first. You want to unmute and ask your question.
Hi, thank you very much for your talk. I have a question about the comparison between the distance probability distribution. I noted that in E. coli cells is only recorded up to 3 microseconds, whereas both in HeLa and in solution it is 4 microseconds. If you were to trim down those ones to 3 microseconds, what would happen to the shape of the distance probability distribution? Would it stay sharp or start broadening?
So you're asking about if this broadening comes from, because it's relatively short deer trace. This is why you're asking.
Correct.
Okay, so in order to cancel the broadening that it's coming from the analysis of when we did the analysis on the deer trace, we took the we took actually the echo decay from here, and we just run, let's say that we use this echo decay equation, and we just extracted here, and we saw that there is no broadening, actually, that it looks like the HeLa cells one. So this just reinforces our thinking that it comes from the protein, and it does not come because of this shortening in that the deer trace that was acquired is just shorter.
Okay, perfect. Thank you very much.
Thank you.
Svetlana, you have a question as well? Feel free to.
Yes, hello, can you hear me?
Yes, yes.
Yes.
Okay, I have a question. So when you interpret also if we stay here, the changes that you see in E. coli, that your distance distribution is broader, and also together with the fact, as you were discussing, that you see much broader CW spectrum, which you could also not reproduce by mimicking in cell environment with viscosity and so on. How do you know this is not just aggregation? Because aggregation is one of the major problems of electroporation, and of course also between different cells, as membranes are different. It can be maybe E. coli have more aggregates, and this would could make your spectral shape broader, and also the distance distribution broader, or maybe also some protein sticks to the membrane. So how can you say or exclude that this comes from the aggregates?
So the reason that we were sure that it's not aggregation is because eventually you get the maxima at the right position. If it was aggregation, it was either you could not see, I mean, the spin labels were too close to each other, and the T2 will be reduced very fast, and you can not acquire a deer. So the thing that we managed to acquire up until 3, that it's relatively very good for in-cell studies. And again, we get a maxima at the right position that we also managed to achieve comparing to the in solution and HeLa cells. And also in HeLa cells, it was examined previously in a previous work. So that make us confident that there is no aggregation. I cannot confirm that in 100%, but I know that the ones that we do observe are probably in a structure that is a bit different than what we see in solution, but it's not aggregating.
But you also don't know the localization in the cell, so it can be you are blind towards this, right?
No, but when we talk about the delocalization of the protein, then you can investigate it using a fluorescent labeled PpiB inside cells.
Yes, but inside mammalian cells, you cannot really localize them in E. coli because they're too small with fluorescent dyes, right?
Yeah, this is true.
So you don't know if maybe some are in, I don't know, in periplasm, or some maybe in inclusion bodies, so it can be everything possible. So you're really blind in a sense where this broadening coming from. Because it can also be mislocalization. It's also often a problem of electroporation, which for mammalian you can visualize, but with E. coli, it's difficult.
It's very difficult, so, yes. So this is another thing that we were trying to do. We did not really manage to prove that, because again, you need the microscopy in order to perform that. So I do not know, but anyway.
But have you?
Sorry.
Sorry. No, but just out of curiosity, have you maybe tried to lyse your E. coli cells after, and try to maybe quickly then separate membrane and lyse it and analyze afterwards?
Exactly. This is what I was thinking to do, because this is something that we are doing here for other protein, and in order to see if it got into the inclusion bodies, we can actually just lyse the cell, and then you can extract between the inclusion bodies and all the supernatant, and then run it on a different wells using SDS page analysis, and you can see if it's there or not. Actually, this is a great idea. I can perform that because I'm doing it on basically basic.
Okay, but I just wanted to ask if you have done it for this particular protein. Not yet. Okay.
Not
right.
yet. Thank you.
Okay, Lewis, you want to go ahead and ask your question?
Yeah, thank you. Thank you. Hi, Yasmin. I very much enjoyed this. This is somewhat removed from my day-to-day thought process, so it's always nice to see another form of magnetic resonance. I just have a couple of questions. You had that slide where you looked at a number of proteins that you had added to your protein to try to mimic viscosity and the like, but you were at presumably much lower concentrations than you would
exactly
in cells. So well, here you have 300 mg per ml of phycol, but that's not really a protein. So well, you have 200 mg per ml of BSA. So I take that back because that's more in cell type concentration. So your feeling is that somehow in E. coli there's some interactions that are occurring that are not viscosity related, or at least not similar to what you would get with BSA, which causes some sort of rigidification. But is it surprising to you that you see a distinction between sort of a mammalian cell system and an E. coli cell system? I mean, you know, these molecules tend to be fairly, I would imagine, promiscuous in their interactions. And so you have such a wide variety of proteins in a mammalian system. Why would it be that you wouldn't see these sorts of interactions there, do you think?
Thank you for this. It's actually a good question. So the first thing that you said, yes, our assumption was that we did not manage to mimic the cellular environment. First, also, this is also related to the cell lysate experiment, because again, we don't have the same complexity and crowdiness as the cell. And this is why we suggested, this is why maybe we cannot mimic it, but the conclusion from here is that you need to do in-cell if you want to really understand what is happening. And for the second question, we thought that the reason, or the concept of the quinary structure is founded on the fact that there is a unique features of the that is co-involved together with the protein and all the other protein in its vicinity in the E. coli cells. And this is what makes this interaction on the surface of the protein. And this is actually the quinary structure. So the reason that, so when we saw that we do not see this broadening in a non-native, and this is why I highlighted that it's a non-native cellular environment, we did not see the same change. It was really surprising, and also support this concept, this elusive again concept of quinary structure, because this is exactly the definition of quinary structure. That only in the native environment of the protein we should see this quinary structure, and again, it's because there is a specific binding partners that are in. Also, if we are talking about the concentration of some proteins, or because we know the affinity is affected also by the concentration that is there. So all of these affect and transient interaction should affect the quinary structure. So I hope
you know that you don't have any unfolding of your protein. I mean, often when you put proteins in a highly proteinaceous environment, like in a phase separate type environment, you get destabilization of the protein. Could the broadening that you see be a reflection of that? That you have some sort of, I mean, I guess it could, and that's what you mean when you say you have some interactions. I mean, presumably the interactions are going to be many-fold, but probably fairly transient, I would assume.
exactly.
And then one last question, your dear trace, it looked like in solution you had a little peak at a larger distance, and I wasn't sure how to interpret that.
Yeah. So we actually, everything that you see here, we of course have a biological repeats, and in others deer trace you cannot, there is nothing there. So basically when you validate it using deer analysis, this is the software that we used, these peaks are getting deleted almost. It's in the range of where it's not verified. Let's call it like this. So what you can understand from here is that you have this main distance distribution. The main distance is this 4.5. So again, the larger distance, the longer distance, we did not see it, we did not observe it in other traces that we acquired. So I guess this is something maybe specifically in this sample.
I see. And just one final quick question. In terms of the broadening, again, I guess it doesn't have to be proteins. It could be nucleic acids. It could be membranes. I mean, could you do sort of in vitro controls where you added membrane or you added DNA and you actually repeated?
No, we did not do that, but we thought that because we add the cell lysis, then maybe we can see, and this is something that we plan to do, just to we can see if there is some change, if we're gonna take again this double labeled gadolinium variant, and we're gonna just to understand if we see the same deer trace like in solution or in HeLa cells, or we see some broadening. And this could be something that is very interested to see. But as we saw before, the cell lysis does not mimic well the cellular environment. So this is why we did not measure it yet.
Thank you.
Thank you.
Marlene, you have one more question?
Yes, if possible. I was just wondering because I think I didn't really get what you said. Could you explain me the difference between a quinary structure and an interaction region? Or is it the same thing?
And the
interaction region, protein-protein interaction region.
Okay, so about the quinary structure, we are talking here specifically about the total effect that are existing inside the cell that can affect this. So it's not only protein-protein interaction. You can also have the crowding effect and the stickiness that can happen too with the other micromolecule inside the cell. You have the it's like everything that you can think of.
Okay, so it's like something which inside the quinary structure you have all sort of interactions that you can.
So it's not inside. It's basically here we are talking about another layer. If you, as I said, you go from these 3D structure that are interacting with each other, then as you said, you have the interaction space, and here we're talking about the surface interactions. Now, I imagine this like in PpiB when you start to see the surface and not the structure itself, then you can see this transient interaction. So it's like another level of it's different.
And what would be the time scale of that transient interaction? Do you have an idea? Is it known?
I cannot nominate a number, but it should be something that is relatively fast, because again, it's not. I'm not sure if somebody prove it.
No,
but it's
a very naive question.
No, it's actually a good one. Maybe we should focus on that.
Okay, thank you. Thank you very much.
Okay. I don't see any more hands raised. Going once, going twice. Okay. And I think we thank Yasmin once more for such a nice presentation and for fielding almost 30 minutes of question and answer session. That must have been challenging.
But it was
to say the
great
least, but
question,
since-
so thank you. You give me a lot of stuff to think about and to continue.
Of course.
Thank
Right, the
you
work
very
is never
much.
done.
Yeah, never.
Okay, with that, we close the session, and I hope you will all come back in 2 weeks, where we will have the next speaker in our program. So look out for those emails, and I'll try and be a bit more careful about timing, or at least the descriptions of time zones in our next email blast. So thank you again, and I hope you all enjoy the rest of your day, morning, evening, wherever you are in the world.